Good evening, everyone, and welcome to this special episode of the UT Southwestern Science Cafe about understanding COVID-19 vaccines. I'm Jenny King, Director of Public Affairs here at UT Southwestern Medical Center in Dallas. On behalf of my colleagues, Dr. Trish Perl and Corey Tovian, thank you for joining us. We are presenting tonight's Science Cafe in response to strong community interest in learning about COVID vaccines. In fact, you all are the largest cafe audience to date, so we thank you. To facilitate Q&A this evening with such a large audience, we are asking Dr. Perl questions, which many of you submitted in advance. We hope her presentation and Q&A session will provide each of you with valuable insight and education about the newly released and upcoming COVID-19 vaccines. Before I introduce Dr. Perl, I have additional UT Southwestern COVID information to share with a number of links that we will also share in the chat box. First, we just wanted to make sure you know we have extensive information and resources on our website about COVID, utsouthwestern.edu/COVID-19. We also have a number of COVID-related medical blogs at utswmed.org/medblog/COVID. We have extensive research underway at UT Southwestern, more than 250 projects, many of which are collaborative with our domestic and global colleagues. And one of the things I'm honored to work on is the DFW COVID-19 prevalence study, which we invite you to join as a community member or an essential worker. This study is presented by UT Southwestern and Texas Health Resources. It encompasses individuals in Dallas and Tarrant counties, 44,000 people, in fact. It's a randomized convenience sample, and data will inform future health public policy. We invite you to go to our website at utswmed.org/COVID study to see if you qualify for free COVID testing. And if you think you work at an essential workplace, which really means you are not working from home and you are either working with coworkers or interacting with the public, please email Corey and me at our public affairs inbox, and we will give you more details about this program. Before I introduce Dr. Perl, I have a couple technical matters to review. We are recording this program and live streaming it on our Twitter page. Due to the timely nature of this topic, we have invited media to listen in to the cafe. Additionally, we will be sharing this recording on UT Southwestern digital channels post-event. If we do have any open time at the end, we will take additional audience questions from the chat box. We will conclude no later than 8:15 p.m. this evening. And finally, just a reminder, while we can't answer personal medical questions, we really appreciate hearing from you with your general questions about COVID-19 and the vaccines. So we thank you. And now on to the main event, Dr. Perl. Dr. Trish Perl is Chief of the Division of Infectious Diseases and Geographic Medicine, as well as a J.P. Sanford Professor in Infectious Diseases here at UT Southwestern. An accomplished infectious diseases physician and researcher, she is also world-renowned for her work in this field. Likewise, Dr. Perl is experienced with management of past international healthcare crises. She is past president of the Society for Healthcare Epidemiology of America and has consulted with international governments on strategies for infection control. In addition, she has served on advisory panels for the Institute of Medicine, the CDC and WHO, and consulted with the NIH. Dr. Perl received both her undergraduate and medical degrees from the University of North Carolina at Chapel Hill and a Master of Science degree from McGill University in Montreal, Canada. She completed her residency in internal medicine at McGill and a fellowship in infectious diseases and clinical epidemiology at the University of Iowa. We are very lucky to have Dr. Perl on our team here in Dallas. She has been monitoring COVID-19 since she first learned of a mysterious contagious disease late last year, and she is on the collaborative leadership team for our work at UT Southwestern related to this virus. Dr. Perl, welcome to UT Southwestern Science Cafe. The virtual podium is yours.
Thank you so much, and welcome everybody. And I hope you all have a beverage and that I can entertain you for the next few minutes, and then we can ask some questions. So one of the first things we need to say about certainly the COVID vaccines is I am telling you what I know right now. I'm also going to tell you that it could change tomorrow. This field is moving so quickly that it really behooves each and every one of you to keep abreast of this or to reach back out to us so that we can answer questions as they develop. So we're going to do a quick tour, a little bit about vaccines, a little bit about COVID vaccines, and then a little bit more in depth about some of the candidate vaccines and where we are in the approval process and the rollout to attempt to mitigate the current pandemic. So, the first question, and we always have fun with these, is historically, what two medical innovations have saved the most lives? Am I supposed to be reading the answers?
You can if you'd like, or we could just let people.
So clean water, antibiotics, preoperative cleaning, such as sterilization of the skin, sterilization, vaccines, or medical imaging.
I'll give it about another 10 seconds and then I'll go ahead and share the results.
While she's doing that, you can see too the first isolation gear from the bubonic plague. So this is about 1364, and the initial mask. So this is the first we know of masks. And then below you can see Jenner giving the smallpox vaccination. Oh, so this is terrific. You guys get A's. So the two interventions were actually vaccines as well as sanitation and clean water. So those really have been two of the most important interventions and actually remain important interventions in what we need to do to continue our work in terms of mitigating illness and the spread of infectious diseases. That being said, I will tell all of you that no one's actually looked at hand hygiene and washing your hands in isolation, so that may be right up there. Okay. When it is available, are you planning on taking the COVID-19 vaccine? Yes, no, or undecided? Excellent. You are above the national average. This is very good. All right, let's find out. The next question is, if you answered no, please provide your rationale for not wanting the vaccine. I do not think that COVID-19 is dangerous. I am unsure of the science. I am uncomfortable that it was approved so quickly. I have religious objections. I do not believe in vaccines. Other reasons, and please list them in the chat box. We are very interested. Oh, this is very interesting. So the most common reason is I'm uncomfortable that it was approved so quickly. So hopefully we can give you some additional comfort at over the next few minutes. Okay, so let's go ahead and get started. So I want to start off by telling you a little bit about the SARS-CoV-2 virus. This is the virus that causes COVID. You can see that this is a beautiful virus. It looks like a crown, which is where it gets its name, couronne, or the crown. And you can see those beautiful flower-looking projections that are what we call the spike protein. Based on work that was done with the SARS vaccine, the SARS epidemic in 2002 and MERS-CoV in 2012, and what we learned not only about the immune system response, but also about how consistent the spike protein was across all coronaviruses, as well as developed several vaccines that were implemented in certain situations, it became evident that the spike protein would be the most likely target for any kind of vaccine that would be a potential candidate for COVID-19. And so on the right, what you can see is this receptor binding domain, which is not only important in terms of how this virus attaches into the respiratory epithelial cells, but also because of its consistency across the coronaviruses, also remains the target for antibody ultimately that is what we want to see with vaccination. I also just want to quickly tell you a little bit about your immune response. And I know this is a very complicated slide and it's 7 o'clock at night and you're not that interested, but there are two types of immune responses. Both of them are important in vaccines, but it's actually the adaptive immune response on the right that ends up being the one that we're most interested in. But just to go over a little bit the innate immune response, what's really important is the viral RNA, which you can see in the left upper hand corner, is important in terms of stimulating some of the pro-inflammatory cytokines. And ultimately, these interact with mRNA and help it export, if you will, from the nucleus into the cytoplasm. The other component of this is the adaptive immune response. And this is the one that I'm going to end up talking about a little bit more. This is important because the spike protein actually is going to be important in stimulating the T cells as well as B cells in terms of developing and producing antibodies, which are seen right here. The third concept I want to review before we get into this is herd immunity. And so herd immunity is really the indirect effect of immunization or vaccination, and this is why we're doing it. So essentially what you see here is a group of individuals on your left, and most are immunized and healthy as seen in the yellow, but you have two little red guys who are not immunized, sick and contagious. And then you have some blues that are not immunized but still healthy. So most of this population, if it gets immunized, it remains, the spread of disease is then limited. However, if not, you can end up with an explosion among the not immunized and contagious. And so essentially what you're trying to do is cocoon the people who are not immunized and healthy from the people who are not immunized, sick, and contagious. So when we talk about immunization, and sometimes you might hear, well, this person couldn't be immunized because they have acute leukemia. Those would be the not immunized and not sick, and we want to cocoon them and protect them. But with a disease that is as infectious as coronavirus, you actually have to immunize a relatively large part of that population to have an effective cocoon. And vaccination is one way of achieving natural immunity. The second way would be actually natural infection, which is not something that we tend to allow to occur in a disease that has significant mortality in certain parts of the population. All right, so now let's talk a little bit about vaccines. And this is actually true for all vaccines, not just corona vaccines. So there are 5 types of vaccines that we talk about. We talk about DNA and RNA vaccines. What you would know about DNA and RNA vaccines, or not know, these are actually new vaccines, and these are the ones we're going to be talking about today. I'm going to describe this in a little bit more detail, but this uses DNA or RNA molecules that actually then send information to your immune system and tell them to develop the viral proteins and ultimately the antibody. What is fantastic about these is that they're easy to develop and quick to design, and they're very easy to manufacture. The challenge with them is that mRNA is very unstable. And so you've heard a little bit about this cold supply chain. That's the challenge with this vaccine. And this vaccine actually, it actually is a direct descendant of the Human Genome Project. The next type of vaccine is a live attenuated vaccine. This is where we use actual virus or bacteria or organism, and it's weakened. It's passaged through multiple cell lines. And so you have the virus, but it's in a form that it doesn't really cause human disease. It does, however, stimulate the immune system. The advantage is that these are some of our best vaccines. They're extremely robust. The disadvantage is it's not a vaccine we can give to people who have compromised immune systems. Examples of this are measles, mumps, and rubella vaccines, so the MMR, chickenpox. And there are actually some COVID vaccines that are being developed using this formulation. There are then inactivated vaccines where you use part of a virus or a bacteria. It's killed usually with chemicals or heat. These are safe vaccines because the organism is dead. They're pretty easy to make. They do not necessarily engender as much of an immune response, and in some cases have made disease worse. One example of this would be the polio vaccine, the killed polio vaccine. And there are some of the COVID-19 vaccines that are using this formulation. Subunit vaccines are vaccines that use a piece of the DNA of the viral surface or the bacterial surface to focus your immune system on a single target. The advantage is that they really focus on just a single target. They can't cause infection. But again, they may not stimulate as strong an immune response. And we have to commonly add other chemicals called adjuvants to boost the long-term immunity. Examples of this would be pertussis or your DPT, hepatitis B, and the HPV vaccine. One comment I will make about these vaccines, I'm not going to talk about them anymore, is that these may become important later on in several years if we need to do any kind of booster of the current vaccines that we are rolling out. And then the final technology or formulation is something called a viral vector vaccine. This approach really takes a harmless virus and uses it to deliver viral genes into the cells to actually build immunity and form antibody. What's good about them is that the live viruses tend to stimulate stronger immune responses, more so than some of the killed vaccines or subunit vaccines. The challenge with these is to pick a viral vector that is truly safe, and if you don't do that well, the vaccine may not be as effective. The vaccine that has currently been developed using this technology is the Ebola vaccine. And this is the technology that's being used for the AstraZeneca Oxford vaccine. So now I just want to move on and back to our little cartoon and talk a little bit about the COVID vaccines. And again, remind you of some of the details I just went over. So there are some inactivated vaccines in the left upper corner that are chemically inactivated. There are recombinant protein vaccines where the vaccines are composed of the recombinant spikes. And that's usually the receptor binding domain. And then there are viral-like particles that are devoid of any kind of genetic material, but display spikes that also can elicit a response. There are the viral vector vaccines where the virus vector is modified and expresses the S protein, the spike protein. And finally, there are the RNA vaccines, which constitute RNA. These are put into little lipid nanoparticles that I'll just show you in a minute. It develops the spike gene, and then that is extruded and forms an immune response. And then finally, there are the DNA vaccines that contain circular DNA that has part of it encoded for the spike protein. So those are the types that are out there. So let's go into, drill down into the mRNA vaccines. And the reason we're drilling down is these are the two vaccines that, well, actually one has an EUA and one we expect to get an EUA in the next few days. So let's talk a little bit about this. So what happens is you have your coronavirus. You can see the spike protein gene in the middle of this, the little squiggly red line in the middle. And essentially what is going to happen is that that particular this organism will be placed in a lipid-based nanoparticle, and that is to allow this spike protein mRNA to be delivered, if you will, to the cell. That migrates, it then is fused into the cell, which is the middle pictograph, if you will. And what you can see is that when it migrates to this cell, into the cell, the mRNA is then integrated and the machinery of the cell will produce the spike proteins. The various spike proteins that are there combine, and ultimately the cell either bursts or the spike proteins will migrate back out to the surface. And then a cell, which is usually one of our helper T cells, will recognize this spike protein. And then with some help from your antigen presenting cell, activate your B cells. And the B cells are ultimately the cells that are going to develop the antibody that targets the protein. Antibody then mark the virus for destruction, which occurs via this activated T cell. Once this process occurs, your cells may forget about it. But if the virus is then reintroduced into your body, the B cells, the T cells all work together and recognize it relatively quickly. And then you actually jump very quickly to the stage where the B cell starts producing antibody and negates any kind of infection with the virus. So let's talk a little bit about these mRNA vaccines. This is a novel technology, but as I said, it's based on a lot of work that's been going on for the past 20 years. The first component, and the first thing that happened, and this actually happened relatively quickly in January of this past year, is they wanted to find out if they could give the candidate vaccine to people and actually get any kind of immune response. And so what they're doing here is they're measuring something called IgG, which is a way of capturing your antibody response. And so you can see this is a complicated slide, but what I really wanted to show is that there was if you look and compare on the far left, where you have hardly anything with the gray, which is the placebo, and then you look at all of the different dosing strategies for the vaccine, you can see that with each subsequent dose, you either get a large or a smaller increase in the amount of IgG that could be measured. And so that was very reassuring. And what was also important is that the response didn't differ very much if you got 10 nanograms versus 330. And so they knew very quickly that they could give the lower dose of this and have as robust an immune response as with a higher dose. The second thing you can see on this particular slide, regardless of whether you're looking at the top panel or the bottom panel, which is looking at antibody response in a little bit different way, is that it was equally robust in people who are 18 to 55 as well as the elderly. And that's also been very important because for the elderly who are at the highest risk of complications, we wanted to make sure that any vaccine that was developed in a way that it would target one of the populations of most importance. So enter now Operation Warp Speed. And honestly, that last slide is part of Operation Warp Speed. But what was really unique about this process, and frankly remarkable, is that a typical vaccine may take anywhere from 72 to even longer to develop in terms of doing all of the initial work in animals, the work that I just described, as well as the clinical trials. And if you look at the bottom panel, what the group really wanted to do was to have a much more truncated response. So they had 5 months to really create vaccine candidates, 6 months to do large-scale clinical trials, 3 months to really sort out exactly who this vaccine was going to be used in. And simultaneously, they are manufacturing vaccine as they're doing this. So as opposed to the manufacturing process, which takes place, as you can see on the top slide, normally after all the clinical work is done, this vaccine was getting produced at the same time that we were studying it. Just to give you a sense of the landscape, there are 37 clinical trials that are in very early stages where we're looking at safety. 17 that are in expanded safety trials where they're getting tested in larger groups of patients. 13 that are in phase 3 studies where they're doing large-scale efficacy trials looking at lots of different patient populations or person populations. 6 vaccines that have been approved for early or limited use. One of those is in the United States, and none have been approved for full use. So today I'm just going to talk about actually I'm going to mostly talk about the Pfizer and the Moderna vaccine, but what I wanted to show you is the 4 vaccines that are a part of Operation Warp Speed. So you have the Pfizer and the Moderna vaccine, which are mRNA vaccines, and then you have the AstraZeneca and the Johnson & Johnson vaccine that are vector vaccines. The Pfizer vaccine has an EUA. The Moderna vaccine got recommendation for an EUA approval today. The Oxford AstraZeneca is doing their preliminary analysis at this point, and then the Johnson & Johnson vaccine is in clinical trials. Why we have so many different formulations and what's important, the Pfizer vaccine requires very, very cold storage. And so that is going to be one that's more difficult to use in rural areas where they don't have sophisticated freezers. The Moderna vaccine it requires a cold storage supply chain, but it is not as unforgiving, if you will, as the Pfizer vaccine. And so that has much more possibility in terms of distribution throughout United States, as opposed to just in large centers. And then the Oxford AstraZeneca vaccine doesn't have any of these cold storage requirements, and maybe a vaccine that is going to be very useful in parts of the world where you don't have the cold storage facilities that are needed. And then finally, what's lovely about the Johnson & Johnson vaccine is you only need 1 dose. And so that's going to also be an advantage. Oh, excuse me. All right. So I've already told you about these vaccines. I just want to quickly go to the efficacy results, which we're going to look at in more detail. But overall, the Pfizer vaccine had an efficacy in terms of preventing a COVID infection of 95%. The Moderna vaccine is 94.5%. And the Oxford AstraZeneca vaccine is 70% efficacious in one part of the study. And then in a different formulation or different dosing strategy, it was up to 90%. So I would expect, and in fact, in all of these, there are additional studies that will be developed relatively quickly. So I'm going to go into a lot of detail about the Pfizer vaccine. Why? Because the paper has been published and we have a lot more information we can share with you. I think that if we were doing this talk in a week, we would be able to do the same with the Moderna vaccine. But the first thing I want to show you about these clinical trials is they are ginormous. These are almost 44,000 patients. You can see that when you look at men and women, they're equally divided. And this is what we call randomization. So if you look at the race and the ethnicity in both the arms, you can see that it's almost exactly the same, which is exactly what you want with randomization. The other thing that I'd like to point out is that we really have a nice representation of different ethnic groups, and especially the ethnic groups that have been at high risk of complications, specifically African Americans and Hispanics, as well as they did have some of the South Pacific ethnicities also. The second thing to notice with this particular table is that they had a relatively large number of people, not only over the age of 55, but from 65 to 74, and even in each arm, almost 1,000 persons who were over the age of 75. And then finally, we're not showing you the data, but almost 50% had some kind of medical condition or what we call a comorbidity. So we're capturing those people with diabetes, obesity, that we know are at high risk of complications. What did they look at? What were they testing? What was important? So we call that a primary endpoint, and then we have a secondary. So we always have the, what's the most important question? And then we have some secondary questions. So starting 7 days after the second dose, they wanted to know whether or not the group that had received vaccine had lower numbers of new COVID infections, whether or not they had had a previous COVID infection. So irregardless of if you've had COVID, does the vaccine actually give you additional protection? And then the second endpoint, the secondary endpoints were whether or not it decreased infection actually 14 days after the second dose, then whether or not, and this is very important, it decreased severity of infection, which some vaccines actually don't totally prevent it, but they just decrease severity. And then, as we mentioned, how did it work in some of the subpopulations of vaccine? And just because everyone's going to want to know, what are they defining as a COVID infection? And so what you can see is you had to have one or more of these symptoms, and you had to have a positive PCR. Okay, so what did they find? They found this vaccine, as I mentioned earlier, prevents almost 95% of new COVID infections. And what's fantastic about this particular vaccine is that the efficacy was similar in different subgroup of ages. If folks were obese, if they were male or female, in the different ethnicities and races. And so what we really saw with this was consistency across all of these subgroups. And so as an epidemiologist, that to me is very important. If I see it's consistent, then it makes sense. So I think that's a very good sign. A couple of other things. Did it work in preventing severe infection? And what you can see here is that it appeared to really decrease the risk of severe infection. So it was about 66% effective in decreasing the risk of severe infection. And this is again 7 days after the first dose. And then when they looked at the entire population, everybody, whether or not they'd gotten the vaccine 2 days ago or 6 months ago, overall they had an efficacy of 88.9%. So really very, very strong results. Now, what about if you just get one dose? Does it work? And here are the data. And so if you'd gotten one dose, but not two, it's 52% effective. If you'd gotten your second dose, but not quite at 7 days, it's 90% effective. And then if it's over 7 days after your second dose, it was, as we said earlier, 95% effective. So even with the first dose, we are getting some vaccine efficacy. This is how it looks over time. And I show you this. You can see the patients who received placebo. Along the y-axis, this is, how many cases of COVID they had. And you can see those that received the vaccine. There were some who were probably incubating vaccine very early on in the first 10 days or so, and may have actually really not been testing the efficacy of the vaccine. In the insert, in the upper insert, I just really wanted to show you that this split occurs around day 10 to 12. And so we're actually getting immunity pretty early with this vaccine. So very exciting. All right, so what about safety? That's the other question everyone's asking about. So here is what Pfizer and Moderna's looks very much like this. So what they did is they had a vaccination period where they were actively surveying people to determine whether or not they had any side effects. And then they started doing it at 1 month, 6 months, and then the plan is to follow these people for 2 years. So what did we see? So reactogenicity means you had a side effect, a response. And the first thing I want to tell everyone is you want to have a response. And so what I am showing you, these mild side effects are important. I also will tell you personally, I got my COVID vaccine yesterday and my arm hurts. So you can see redness, swelling, and pain at the injection site. What I will be pointing out is that it tends to be more pronounced after the second dose. It's not quite as evident here, but these are just sort of local reactions. And this is within 7 days of getting the vaccine. Now, when we look for systemic reactions, this means fever and headache and chills and fatigue. And so you can see that a lot of people had fatigue and headache, diarrhea, muscle pain. What I'd like you to look at is the lower panel in the placebo. And so you can really see how many people who got placebo also had very similar side effects. So that differential is, at least for fatigue and headache, about 10% more in the group that had the vaccine. This is why placebo-controlled trials are so important, is that many of us have this idea that when you get a placebo, you're not going to feel anything, but it just really tells you what the mind can do to trick you. The other important point is that while there were some people who had relatively high fevers with the little gray lines here, they were very, very few. So most of these were what we call mild or moderate side effects. Here's what it looks like after the second dose. Now this is where it becomes more pronounced, and you can see actually that here we had a lot less complaint in our placebo group, and there does seem to be much more of a response that, again, is what we want to see. We know your body is reacting to this vaccine. So this is not something that concerns us. This is what we tell you to expect. And if you develop it, we can offer, some Tylenol or some non-steroidals to make you feel better. And this is a different way of looking at it, but just really to give you a sense of how common these were. So each of these little dots represents 8 people. And so you can see how many people got sick with COVID and those that received vaccine versus placebo. So 8 versus 162. And then you can get a sense of the adverse reactions after the second dose in these different groups. What about more severe side effects and any of those over long term? What was noted? So there was some long-term complaint with with soreness at its site where the vaccine was given. And then you can see that there are a myriad of other different types of side effects that were reported to the sponsor of the clinical trial. What I would like to point out is what we call hypersensitivity or allergic reactions. There were 137 in the vaccine group and 111 in the placebo group. This is from the FDA's data analysis of this data. Interestingly, and people haven't commented on this, lymphadenopathy, which means lots of lymph nodes, so just swelling of your lymph nodes, 64 in the vaccine group and 6 in the placebo group. This resolved usually around day 10. And Bell's palsy, 4 in the vaccine group, none in the placebo group. This was not called out because this was felt to be consistent with what we would see in the general population. All right, how about serious adverse events? So here are the serious adverse events. There were some myocardial infarctions that were reported. There were several strokes that were reported. And interestingly, appendicitis was reported in both groups, but none were deemed by the FDA, not the sponsor, to be related to vaccines. There was one shoulder injury. We mentioned the lymphadenopathy that were thought to be related to the vaccine, and there was a ventricular arrhythmia, so an abnormal heart rhythm that was not thought to be related to the vaccine. In terms of deaths, there were 2 in the vaccine group and 4 in the placebo group. None were attributed to the vaccine. One was a cardiac arrest 62 days after the second dose, and one was from arterial sclerosis 3 days after the first dose. What about the Moderna vaccine? So this is from the FDA data that was released at the meeting today. And you can see that, again, this vaccine looks equally impressive in terms of its efficacy, with an overall efficacy of 95%. And, importantly, some of these people have been reported or followed at least 3 months. So now I want to kind of step back a little bit, because I've just showed you all these side effects, and I want to compare this to a couple other vaccines that many of you may have gotten. So on our far right, we have placebo, and this is how many people report any kind of side effect from getting placebo. So you can see overall 13%. When you just take one of the flu vaccines, you can see how many people report a side effect, 14%. When you look at the Pfizer COVID vaccine, it was about 26%, with 83% having local reactions, and then the rest being systemic. If you look at the herpes zoster vaccine, or the varicella zoster vaccine, Shingrix, which many of you may have gotten, that side effect profile is about 38%. So just to give you a sense of where we're talking relatively. What do we know about pregnancy in this vaccine? Well, as in most clinical trials, if you were pregnant, you were excluded from the trial. However, 23 people got pregnant during the clinical trial, 12 in the vaccine group and 11 in the placebo group. That's as of November 14th. And they received the vaccine sometimes within 30 days of getting the dose, or 30 to 40 days after getting the dose. So we sort of have a spectrum of very, very early to very late. Overall, in the group so far, there's been 1 miscarriage and 1 retained products of conception, both in the placebo group. 9 of the pregnant women withdrew from the study, but they were equally split between the 2 groups. The Society of Maternal and Fetal Medicine has looked at these data in detail and recommends that this become a shared decision-making process, but that pregnant or lactating females should not be excluded from vaccination when allocated, especially if they have risk factors. So what's going to happen now? So there is a lot of surveillance that is going on to look for adverse events. This happens with every vaccine. There's active surveillance with an app called vSafe. I enrolled myself. There is also the vaccine adverse event reporting system, which is a passive surveillance system where you can report what you think is an adverse event. And what you can see is both the VA and the DoD contribute to that. The CDC also will sometimes receive its own report. And then you can see that there are also some other different surveillance systems, and all of this is going to get integrated or concatenated, and ultimately there will be a large database that will really be able to look long-term at whether or not there are any long-term consequences associated with this vaccine. So we're gonna stop me talking and we're gonna ask you a question. After hearing all the data, are you planning on taking the COVID-19 vaccine? Please vote yes, no, or undecided. Oh, okay, I got another 3%. Excellent. Or maybe 6%. All right, I'll have to give this talk another few times. All right, so now we're going to turn over to questions, but I just want to remind everyone that just because we have a vaccine, it doesn't mean we can stop not wearing masks, not doing the social distancing, and not washing our hands. It is going to take us a long time to get all the people vaccinated that we need to get vaccinated and to get that herd immunity that I was talking about. So we have our work cut out for us. We still need you to hang in there. We are asking you to hang in there. And just because I wanted to embarrass myself, I am showing you when I got stuck and that little grimace on my face when I got my vaccine yesterday.
Thank you, Dr. Perl. That was fantastic, and I'm glad we all got to learn a lot from you. I am going to move into questions as fast as possible because I know we are coming close to the end of our time, and I'm going to start with the questions that were actually emailed in to our public affairs box. So our first question is, can you still contract COVID-19 if you get the vaccine?
So that's what the vaccine efficacy is really about, is are you going to get COVID-19? And so what we are saying when we say this is 95% effective, it means it's going to reduce your risk by 95%. So the answer is that yes, you can probably still get it, but the risk or that chance is really, really low. And then the good news is that if you do get it, it may be that it's not as severe and it would prevent some of the severe consequences.
Thank you. Our next question is, if you receive the vaccine and then are exposed to COVID-19, can the current test distinguish the antibodies in your system from the vaccine versus a new exposure?
So that's a great question. And the answer is that what we routinely measure to see if you've had a COVID infection, no, we can't. We would not be able to differentiate whether or not this is infection versus vaccine. However, if you're part of the clinical trial, or if you're in a very sophisticated research lab, you actually can get antibody-specific testing done and determine whether or not it's a vaccine strain immune response versus a natural infection immune response.
Thank you. Our next question is, do the vaccines use any chemicals we should be cautious or aware of?
So in general, people, we try not to put any chemicals that that are harmful to anyone in vaccines. I will tell you very frankly, I looked at the list yesterday and there was nothing that struck me as worrisome. But, you know, I think that you have to look at it yourself. And it certainly doesn't have any of the adjuvants or some of the other chemicals that we've seen in other vaccines that have concerned us. So, for example, there's no thimerosal, there's no neomycin. So some of these other compounds that have been in other vaccines.
And this is a really important question. I think a lot of people want to know, if you've already tested positive for COVID-19, should you still get the vaccine?
Right, so that's a great question also. So currently the ACIP, which is the advisory group to the CDC, is recommending that even if you have had a COVID infection, that you get vaccine. There is a kind of caveat to that in that it may be that you want to wait 3 months, especially now when supplies are low, because you are going to have neutralizing antibodies, which are the antibodies you want to have for probably 5 months is what we know at this point after getting the infection.
So staying on that for a second, if a patient received convalescent plasma or one of the new antibody treatments, are they eligible to get the vaccine as well?
So that is actually one of the things that's really important is if you had COVID and you got convalescent sera, you should absolutely wait 90 days. If you got one of the COVID-specific antibodies, either the Lilly or the Regeneron, you should also wait at least 3 months. And the reason is that we are worried that they would interact and decrease the immune response that you would generate as an individual. So we don't think it would be quite as effective in you.
So moving on to some of the concerns we've had, is there any data related to the vaccine and pre-existing conditions such as depression or anxiety causing more intense side effects?
So there was nothing listed in the clinical trial data or in the documents that were given to the FDA that suggested that. And, you know, we're just really beginning. We're like day 3 or 4 of doing a lot of vaccination. So the real answer is we don't know. But what we can tell you in the first, 80,000 people that have been tested, there's nothing that's been flagged for us to warn you about.
So staying on those pre-existing conditions, is the vaccine safe for people who have autoimmune diseases or other conditions such as high blood pressure and diabetes?
So people who have hypertension and diabetes should absolutely be getting the vaccine because they are at much higher risk of complications. Autoimmune disease, people were included in the clinical trial. The current recommendation is that almost everybody should be getting this vaccine. There are a few highly immunosuppressed people who are getting special kinds of bone marrow transplants that we think need to wait. But honestly, any kind of rheumatologic disease, any kind of immunosuppressed individual with cancer, whatever, should really be getting vaccinated. And we're recommending it for everyone.
And that leads into our next question, which you mentioned about cancer patients. Do we know if any of the trial participants were cancer survivors or current cancer patients?
So I don't know that they called out that specific group, but they were eligible to be enrolled. And the way it's phrased, I think that there were, but I can't answer that Specifically, what I can say is the fact that the ACIP is recommending that we give it to people who are on medications that would immunosuppress you or have conditions that would do it suggests that the answer is yes.
So with the Pfizer trial, they enrolled ages 12 and up, but they only approved for ages 16 and up. Can you explain why the change in approvals from what they tested?
Yeah, so I didn't listen to the meeting. I'm going to guess, this is my guess, is that they probably didn't have enough individuals between age 12 and 16 to determine whether or not it was efficacious. I also know that Pfizer is starting a clinical trial in pediatrics. This is a group that we all are very interested in getting additional information in. And so I think that there will be a lot of data forthcoming, probably in the next 6 to 8 months.
So staying on the trial for a moment, were study participants purposely exposed to COVID-19 during the phase 3 trial, or were they just about their normal lives and tested for antibody response?
So they were not purposely exposed, but they clearly were exposing themselves because they got it. And, so I think they were just doing their normal activities and got COVID.
So moving a little bit into the actual makeup of the vaccine, with the mRNA vaccine, does it kill healthy cells in its normal process? And if so, how is this limited?
You know what? I don't know that I can
Dr.
answer
Perl.
that question. That's a great question. I mean, it is a self-controlled process and there is only so much that goes in. And we do have regulations, but I am not an immunologist and I apologize. I can't answer that question.
No, completely understandable. We appreciate you saying that. For the actual Pfizer and Moderna vaccines, what is the reasoning for why one really has to be stored at that -80 versus -20? And if it is not stored properly, is there any issues towards human health?
Yeah, I don't know that any of us are doing that experiment, to be very frank. I mean, this is a precious substance, and so everyone's trying to play by the rules. But what I can tell you is mRNA is very unstable. And so that's why it has to be frozen. And that's the disadvantage of this kind of formulation. The advantage is we can make it really fast because it's easy. It's relatively cheap, but it's an unstable product. And that's why I alluded to the fact that it may be that the Pfizer product is, something that is disseminated at places like UT Southwestern, whereas the Moderna can go into places that don't have these -80 freezers, et cetera. In terms of what happens, you have about 6 hours after it comes out to give the vaccine. And right now, there's enough demand that everyone is going through the vaccine and we're not doing the experiment of throwing out any of the vaccine. I am guessing that as we get a little bit more experience with this, there'll be more and more information. I mean, even in the days coming up to when the vaccine was delivered on Tuesday, the guidance was changing in terms of, how many days could we keep it out? And it turns out with this vaccine that you have 15 days in the containers that it's shipped in if you don't have a -80 freezer. And so there are some capabilities, or there is additional latitude that the manufacturer is looking at to try and see if they can make this vaccine more available to centers that aren't not as specialized.
2 more questions for you before I pass back to Jenny. But a lot of people are dealing with family and friends, relatives that are really just against, getting the vaccine. Do you have any suggestions on what we can say to people to encourage them to go out and get their COVID vaccine?
Yeah, so well, I can say a couple of things. And I mean, first of all, I think we're seeing more and more vaccine hesitancy, which is really unfortunate because as we pointed out in the first question, it is literally one of the things that's changed the mortality rates internationally throughout the world over time. And, you know, the first vaccines were really introduced in the late 1700s is when the smallpox vaccine. So it's not like we've got years and years and years of experience, and most vaccines didn't really get introduced until 1900. So anyway, so that's the unfortunate part about this. So what can you do? So some people are hesitant to do it because they just don't know. So hopefully I have a whole bunch of ambassadors now that have a lot of information that they can take back to their families. So that's one thing. I think the other thing we have to do is really understand why is it that they don't want it? And, you know, sometimes it's just the right person hasn't told them to get it. You know, sometimes it's their doctor that needs to tell them that. Or, you know, somebody like me needs to tell them it's okay. The third thing I can say is more of a personal comment, and that is that I was as skeptical as everybody about this accelerated timeline. I mean, my job is to protect people. And I am stunned at the data with this vaccine. And I think the fact that the Moderna looks almost as good and is very similar is telling us something about this technology. So, you know, that's, I think, the final thing. And we all are basically going to have to be really the ambassadors to get this out. And this is very much going to be like what it took to get people to start using car seats, which, you know, took a long time to get people to buy into car seats or wear seat belts or, you know, not, you know, you can drink, but you can't drive and drink, you know, so I think that this is just one of those things that it's going to take a village to really get that message out, but we also have to understand what it is that makes people hesitant.
And my final question to you, less about the vaccine, but what do you feel from an infectious diseases expertise about the projection of COVID-19? Are we going to see it coming back every season? Is it going to be like the flu?
So what I can tell you is that coronavirus is a seasonal virus. There are 4 what we call endemic coronaviruses that happen every fall. So if this follows that pattern, it will come back. You know, I think this could be like flu. And this is the 1918 event with coronavirus that we are experiencing right now, where it gets introduced into a community. The good news is that unlike 1918, we actually have a vaccine to add on to the masking and the social distancing and the hand washing. But I think that right now, most of us suspect this will be seasonal. We don't know how long it's going to take before we get not only immunity in the older population, but even in the younger population. It may be that as this becomes more and more endemic and kids are exposed to it a lot more and the kids aren't really getting very sick, that they end up getting sort of immunized by just natural herd immunity along the way. And it may require both that plus a vaccine long-term.
Well, thank you. This has been absolutely fantastic. And I'm going to pass back to Jenny to go ahead and close us out.
Indeed. Dr. Perl, thank you so much. This was a remarkable conversation. People in the chat box are very complimentary, and I think you have created some new ambassadors, so thank you. We're grateful for your time and expertise this evening. Corey, great job on the Q&A and minding the technical details. Everyone that joined us this evening, we hope you enjoyed the episode of the UT Science Café series. Be looking for an email. We will be sending you a brief survey about tonight's program. We value your feedback. We will be back in January for the winter season of Science Cafe, so please continue to watch your inbox in the coming weeks for registration details. As we close out this evening, from all of us at UT Southwestern, truly let's work together to keep North Texans safe from COVID. Wear face masks, maintain distance when in group situations, and practice good hand hygiene. And it sounds like from Dr. Perl, this is in the long haul. This is going to be for quite a while. For now, we wish you good health, good spirits, happy holidays, and a bright new year. We are adjourned.